A very good afternoon, everyone. I am Dr. Kailan Mingsami. On behalf of Government Selchip College, I welcome you all to the last day of the 5-day international webinar with the theme Science and Technology Mitigating Current Challenges. Thank you for taking the time out of your busy schedule to attend this event, and we hope the past 4 days have been insightful and informative for you all as much as it is to us. So before we further proceed to the program, here's a few announcements for the participants. Number one, if you have any questions to ask the resource person based on the talk, you can drop your question in the Q&A box down below during the presentation, and he may answer your question after the talk. Secondly, we will be providing a feedback form through your email again, and then I request you all to kindly fill up the feedback form. And thirdly, this program is currently live on YouTube, and then the link is it will be put in the chat box very soon. And then lastly, for the participation certificate, their attendance has been monitored in the system during this webinar, and then we know that some of the participants were having a network issue. So we are going to give all the e-certificate to the participants who are attending any 3 days of the webinar. We will be emailing you your certificate as soon as possible. So moving on to our program, it's my pleasure to introduce today's eminent resource person, Dr. Ravendran Vasudevan. He is the postdoctoral research associate in the Department of Plant Science, University of Cambridge. His work involves developing synthetic biology tools to manipulate the plastome, chloroplast genome for the production of high-value compounds and edible vaccine. He has also done other postdoctoral research in the Department of Biochemistry, University of Cambridge, and in the Institute of Molecular Plant Science, University of Edinburgh. He is an expert in plant cell and tissue culture techniques. He has various technical skills involving molecular biology techniques, engineering cyanobacterial pigment, gene editing, and silencing techniques, to name a few. He has published various scientific papers and articles, and currently he is also a laboratory supervisor for 7 PhD biotechnology students. He has also participated in various national and international talks. I would like to particularly mention that he's a friend of our colleague, our colleague, Sir Tomi Lalmal Soma. So thanks to both of them that we will be able to hear a presentation from such a well-versed person who is from our own country, India. So I now give the time to Dr. Ravi.
Oh, brilliant. Yeah, I think. Can I start?
Yes, sir. You can start.
Oh, yeah. Everyone, thank you very much for the invitation. I would be grateful for the invitation. Today, so I'm working at Cambridge. Initially worked with the Department of Biochemistry. Now I moved on to Department of Plant Science. I'm currently focusing on chloroplast biotechnology. Before we discuss about biology, so just we wanted to know how we came from and how we got several evolutionary pathways. So big and about we start to have a galaxy today. So that's the point where we have, that's our galaxy, a cluster of galaxies, and within the cluster of galaxies, this is the Milky Way galaxy. Where do we live? This area, I mean, I could not able to see the fully Milky Way galaxy, and where you see the Earth. And it form the particles combine together form a and then form a ocean. About even first about 2.5 billion for reducing bacteria was formed. And then about 2 years ago, 2 billion years ago, oxygen level of the Earth has increased where the major event occurred. It's called endosymbiosis. And then we have a plant and then animal. Yeah, the endosymbiotic evolution event were proposed by several people. Initially, in 1956, Andreas Himmer proposed the endosymbiotic evolution. He found that the chloroplast undergoes independent divisions. And then followed by his research, Constantine in 1905 from Russia. He proposed that the chloroplasts are independent organism and the plant cells are chimera, which means it has got two organisms. So two independent organisms living together, that's what he proposed. And in Ivan Villian, he proposed that the mitochondria or eukaryotic cells of the mitochondria of eukaryotic cells derived from a bacteria. And followed by Lynn Merkulis, who proposed a beautiful endosymbiotic evolution based on the large amount of molecular data and data. She proposed the endosymbiotic theory. So that's the early cell. The early cells has got a DNA. When after a period, it condensed to form a nucleus. And when the nuclear containing eukaryote absorb archaea or protobacteria to accept it, and then it lives within the protobacteria, lives within the eukaryotic cells, it become a mitochondria. Then the mitochondria containing animal cells engulfed bacteria, which is a photosynthetic bacteria, which is nothing but the cyanobacteria, and it form a plants. That's how the plants get evolved. About 1,500 million years ago, we have a green algae, and then we have a bryophytes about 500 million years ago, and then a vascular plants. This is the lycophytes where it lives along with the Dianosa period, and then a ferns and gymnosperm, and then a flowering plants has got a dicot and monocots. So today I'm going to discuss about synthetic biology. What is synthetic biology? Synthetic biology is bringing technology to biology. So the idea of synthetic biology mainly focusing on molecular biology. So the synthetic biology, you need to modules, you need to design and build and test. Say rice plant, if it's got some disease like a white disease, then you need to understand the pathway, how to get rid of those infections. So you need to understand the pathway, you build the pathway, and then you select a organism to test the pathway, and then to test it, and then you screen it, and then you analyze the pathway based on the data you have, and then you engineer the pathway into the plant. That's the idea. So I'll take you through one by one, actually. So yeah, so we developed a toolkit called CyanoGate, which is a metabolic engineering pathway designer. So the reason why we start with the cyanobacteria are, as I said, it's nothing but a chloroplasts. So cyanobacteria are, and also it's quite important in an evolutionary point of view, as well as in an application point of view. Cyanobacteria are established platform for the production of natural pigments, such as a phycocyanin, anthocyanin, and a phycoerythrin, and alpha-phycocyanin. These are all the biopharmaceutical compounds. So synthetic biology tools for the cyanobacteria or a chloroplast is lacking. So that is the reason we developed a CyanoGate system. The CyanoGate system has got a 46 promoters with a TSS, where you have a truncated version of promoter. You can add your ribosomal binding site and then a terminator to make a construct. So initially we're testing a cyanobacteria called Utex 2973 and later 6803. So that's how, this is the crown for the CyanoGate modular system, how it works. It works in 5 different stages. You have a level 0 parts, because when you take the DNA, you divide them into parts, like a promoter, and then a ribosomal binding site, open reading frame, and then a terminator. So you clone all those individual modules in a level 0, and then you build them into level 1 as an expression module. And then if you want to have a multiplex DNA assembly. You go from a level 1 to level M and then to level P, and you can integrate that into the chromosome as well as into the chloroplast genome. So using this, you can generate gene knockouts in chloroplast or in cyanobacteria, or you can knock in genes. So you can clearly see that's the wild type and that's the mutant. That's the mocked. It's called mocked mutant. You can knock out. Actually, we put the UOFP and then we measure the XFP. Promoters, it's called Anderson promoters. Those promoters are available at genes. So we modified and then we use those promoter because the CyanoGate system use type 2 restriction enzymes. So it uses BPI and BSA because the main advantage of the type 2 restriction enzyme, it will not leave any restriction site within the constructed plasmid. So we tested the Anderson promoter. We measured the strength of the Anderson promoter in one of the cyanobacteria, where you have a dynamic range of promoter. Potentially, these dynamic range of promoters are highly useful for metabolic pathways and designing. So as I said, we have the -35, -10, and then RBS EOFP. These are the native promoter strength. We use some of the native promoter from cyanobacteria, and then we tested this strength in cyanobacteria, and then we noticed that the CPC560 promoter are really highly expressed promoters. So potentially those promoters are useful for further metabolic engineering. And we also measure the ribosomal strength. So because the ribosome are the major component for the gene expression level. So when you alter the ribosomal binding site, potentially you tend to say increase or decrease the production of a protein level. So by altering the ribosomal binding site, we manage to see increase or decrease the production of proteins. So these are all the parameters are essential in order to design a metabolic pathway, because sometimes you need to have a really high expressive genes, so you need to use a high RBS, and then if you want to have a moderate, then you need to use a medium RBS. Yeah, so what we tested actually, because currently we are living in a CRISPR world, where CRISPR is happening everywhere. So there are several versions of CRISPR. Today I'm going to discuss about the Cas9, CRISPR-Cas9, and a CRISPR CPF1. And the CRISPR-Cas9 is a RNA protein mediated gene editing system. The Cas9 is a protein. It recruits sgRNA to the target site, and then it bound to the PAM. We used to call it as a PAM site. PAM site is a NNT, triple N and T, that goes and recognizes to the DNA where you want to cut, and that will cut and then fuse the DNA together. So which means using the CRISPR system, you can generate knockouts. So far, before CRISPR, we don't have a specific gene knockout system for the plants. So the CRISPR is a really useful technique where you can generate gene knockout where you want. So a specific site. So what we did actually, we modified the CRISPR system to work as a CRISPRi. So which is the CRISPRi is a non-catalytic version of a CRISPR system. It will not cut the DNA. Instead of it goes and bind to the DNA, where you cannot see the transcription translation, so resulting that you will not have the protein product. So it silences the gene. It's like a srRNA. So where you have in A, it's a CRISPR-Cas9, and B is a CRISPR interference or DCas9. C is how to design the guide strand for the CRISPR, and D where we targeted the UAFE in multiple sites. The 31 nuclear. When you have different version of the srRNAs, you clearly see transcript of protein. So,
Hello? Hello? Hello, Ravi? Hello? Our speaker is having a technical problem, so let's wait for a little bit. Hello? Hello, Ravi? Hello? Hello?
Uh,
Yes, I can hear you.
um, I don't know. I'm so sorry about that.
Hello?
Hello. Yeah. I don't know why this is disconnecting. If you want the in this the internet connection unstable. So what I could do, I could switch to give me a minute.
Okay, take your time. It's okay.
Just you don't want to fix the yeah, it goes and come back. So just give me a minute and then I will solve it and then I come back. Is that okay?
Okay, sure.
It's not. It should be okay. I mean, if you want, I can continue.
Yes, please. Now you're audible. Hello?
We it's already media system is but it's analyzing. So already mediated. We have a translation according to the srRNA. So the product will be srRNA. The srRNA about a 50 base pairs. So here you target mRNA, get transcribed, and the srRNA.
Hello, Ravi? Hello? It seems like our speaker is having some technical problem, so let's wait for some more time. It's beyond our control, so please bear with us.
This
Ravi, your voice is breaking. Hello? Hello? Hello, Saravi. Hello.
Hello, could you hear me?
Yes, sir, Ravi, we can hear you. So is it fixed?
Um,
Hello.
Sorry.
It's okay. Take your time. It's okay.
Okay. Let's, can I have screen share?
Okay, just a minute.
I think it says other participant is sharing the screen.
Yes, we can see you now. We can see your screen.
I'm so sorry about the interruption. I'm not sure what's going on. Actually, my connection is really speed, but since I'm sure from sometime it may open. So that's why I copied it up and working. Should be okay.
It's
So
it's
as I said, that
it's
we
okay.
are developing a srRNA modular gene expression system for Synechocystis as well as bacteria, where as I mentioned that you have a mRNA and then you have a synthetic srRNA. The srRNA goes and bound to the RBS ribosomal binding site, where you will not have access to the ribosome to recruit, resulting that you will not have translation. So the gene can transcribe, but it cannot translate. So this is we use a different type of promoter, one promoter to express the UAFP, where you see the blue one is the expression of UAFP. These are the two other promoters, which is a light inducible promoter. It's a green light inducible promoter. When you grow the strain under red light, it produces a UAFP. When you grow them under a green light, then you do tend to see decay of a UAFP. So it's like a light switch mediated promoters. And we also developed a CRISPR CPF1 version. The CPF1 is advanced version of a CRISPR system. So where the Cas9 will produce a lot of off targets. So which means, for example, if you design experiment using a Cas9 that will target the gene of interest, that will also target the other region when you select the PAM, then it will select the off. That's what is called off target. It's also select target on some other region of the chromosomes. There is an advanced version of a CRISPR system. It's called CRISPR CPF1, where you can specifically target a gene actually. So we are developing a plasmid for the CRISPR Cas9 CPF1 for the chloroplast as well as the cyanobacteria. The reason why we are developing a CRISPR CPF1 plasmid because most of the prokaryotic organism doesn't have the homologous recombination, non-homologous recombination pathway. So that's the reason why if you want to do a knockout or knock-in cyanobacteria or a chloroplast, you need to use homologous recombination. But for the plants, you don't have to have a homologous recombination. So that's the reason why we are developing CPF1, where you need to have several ligase enzymes to fuse together the cut site. And also we are developing an Operon system because we need to understand that the chloroplast of air plants or the genes are in Operon systems actually. So for example, a single promoter will drive 7 or 8 genes. So it's called Operon system. So we need to have the Operon plasmid system to do a metabolic engineering in the chloroplast. So this is the construction of Operon plasmid. So you have a single promoter, and then you have an individual ribosomal binding site, and you have a multiple open terminator. You combine the single plasmid. So so it works quite nicely in and you test with the modifying produce high value compounds, beta carotene and astaxanthin as so the
Hello.
yeah, I can hear you.
Okay, okay.
I'm so sorry about that.
It's okay,
Going on,
Ravi. It's okay.
but anyway, the main reason why we are focusing on photosynthetic microorganism is nothing but a chloroplast, modern day chloroplast. Spirulina, edible, especially in Asia and Africa. So that's the reason why we are focusing on cyanobacteria. Once we develop a toolkit for cyanobacteria, potentially you can replicate it in. So the cyanobacteria, now I am talking about how to modify spirulina, the vaccines. So in spirulina, potentially I can use the same system for a hair plant chloroplast. The cyanobacteria, this is the photosystem, that's the cyanobacteria, that's the cross section of the cyanobacteria where you have a safe and that's the photosynthetic compound in a cyanobacteria where you have the photosystem. The cyanobacterial photosynthesis is based on phycobilin The phycobilin moves across the photosystem. Phycocyanin. These phycocyanids are highly medicinal values. These phycocyanins are each phycocyanin rod as whether we could be able to put the free forms of phycocyanin in cyanobacteria. How we do? So that's the commercial applications of phycocyanin. You have a pharmaceutical value, food industry value, fluorescent property, and nutraceutical, therapeutic, and cosmetics. So we collaborate with a private company to develop a level of production of phycocyanin. So phycocyanin normally produce a high level in red light. So that's the bioreactor. Good to see the bioreactor. The reactor where you have customized growth chambers where grow phycocyanin and bacteria early a color purify medical grade medical grade knockout or can the mutants to be
Hello? Hello? Okay.
we used a modified version of a CRISPR called CPF1, where you have quite nice colonies, and the colonies we developed. As I said, the proteins are linked by a linker protein, so you can remove those linkers and there are no phycocynin, is supposed to be phycocynin attached to phycocynin C. One you see increase free phycocynin, and then when you remove two linkers, you tend to see more phycocynin. The no phycocynin only using developed for phycocynin.
Hello? Hello, Ravi? Hello?
Yeah, I can hear you.
Okay, okay. Please go ahead.
So not we modify into so industry. So you use, if you know the technique with the strain, you can and you can produce within cells. You just break open the cells, you get phycocyanin, and this is the modified version of phycocyanin, the thermotolerant phycocyanin. So these type of phycocyanin useful for food and assembly cake, and then you can use a potential phycocyanin, and that will give you quite nice.
Hello? 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 Kan speaker hi a signal du angina chalo ma participants te khong ai tikin kan lo shetiam sham sham do ya. Hello? No, ngak le lokila ron lut vele ni turangai.
Zoom meeting,
Hello. As I have mentioned before, our speaker is having a technical problem, so please bear with us. He's fixing it. Let's wait for some few minutes. Okay.
Sorry,
Hello.
I'm a little bit far away from the you guys, so maybe
It's
that
okay.
could be the reason why I got a problem.
Maybe it's okay.
Um, so yeah, so as I said, we could not only produce edible version of spirulina has got edible vaccines. We can also produce lots of other, containing spirulina, because spirulina are edible, and also it's naturally occurring strain. You could, if you could modify it, you can usually use the therapeutic compounds through the oral activity. So yeah, as I said, we so far we got the plasmid, and we got the synthetic biology system to modify cyanobacteria. So that we are moving on to a chloroplast. Why we are targeting chloroplast? Chloroplast is you can do advanced engineering in chloroplast, where you have a multi-gene, because a single chloroplast has got 1,000 copies of a chloroplast genome. That's the most important thing. If you target into the chromosome of a nuclear chromosome, so you have quite a lot of gene silencing and pleiotropic effect, and so many other things. And if you target chloroplast, chloroplast has got a multi-gene and then a gene containment and high copy numbers and no potential pleiotropic effect and no positional effect. So sometimes and then way into a neutral site, sometimes there will be a region called protein. So that's the chloroplast doesn't have a silencing, and so you don't have to have a vector sequence to retain in the chloroplast to produce the foreign DNA. So, and also it's hyper expression. And so what chloroplast engineering is similar to cyanobacterial engineering. That is the reason why we our engineering in cyanobacteria, and we're going to replicate the same to chloroplast. Currently, I'm focusing on Markantia, which is a bryophile, which produce a gemma, and of having a chloroplast engineering, because chloroplast will carry and if I as chloroplast the egg. We target the then potentially foreigners will have the foreign DNA gene, and we have it's called gene foundry. You could you could you the last have gene foundry and synthesis. Okay, so I'm working on genome, read in the chloroplast genome and help, uh, then you can utilize the gene foundry to the chloroplast, use the gene to in high.
Hello. Hello? Hello, Ravi. Hello? As I've mentioned before, our speaker is having a technical problem. So if the presentation stops in between, please bear with us. Technical problem te kan control back til ani thu soi kar la ka lo top deuh zau zau ni phone khong ai tikin participants ten min lo she thiam sham don ya lo hak le lok meyang.
Sorry, I think I believe it's the last slide. You could modify chloroplast genome in such a way you want, and then you can also integrate a lot of other metabolic engineering pathways into the chloroplast that express into in the few. Currently we are developing. We are extracting electric organisms. So once we modify the chloroplast to produce a more output, electricity is nothing but electrons. So when you grab more photosynthesis or higher plants, potentially you can have that's the table. Actually, that's the table lamp using powered by cyanobacteria bryophytes. So potentially you can have your own plant in your home, and then you can electrify the entire house. Actually, potentially you can use a fan, you can use a table and things like that. Oh yeah, thank you very much for the listening. I'm so sorry about that. I don't know what happened. Maybe since I'm a little bit far away from India, so that could be the reason. Maybe it's almost 20,000 kilometers away. Yeah, thank you very much for listening.
Okay. Thank you, Ravi. In spite of all the technical problem in between, the presentation was very good, and it was such an informative presentation. I hope all the audience are enlightened as much as I am. Thank you so much. And then regarding the technical problem, it's all okay, because sometimes things are going beyond our plan. It's all a part of life, so that's okay. And then among the panelists, are there any questions? Panelists?
And I can if you need, I'm quite happy to share the slides so the people can easily have access to the slides because of the difficulty.
Okay, thank you. Thank you so much. Among the panelists, anyone?
Question kavan zot dui o.
Yes, please. Hello? Hello?
Question
The
ka zot
question?
wa ka type lu thei mi lo. Ravi, I have a question, but I can't ask you. Hello,
Yes, sir, we can hear you. Satomi, we can hear you.
Miss
Maybe
Ravi, I have a question. Hello,
Yes, Satomi, please continue. We can hear you.
go ahead.
can you hear me? Am I clear?
Yes, you are
See,
audible.
Ravi, is there any scope of gene?
Hello? Hello? Hello, Satomi. Can you please repeat the question? Your voice is
Is there
breaking.
any scope of gene editing
Can you repeat the question, please?
viral RNA COVID-19 using the CRISPR technique?
No, sorry, I mean, your voice is breaking. I cannot able to understand the question properly. Maybe anyone can
Okay,
repeat the question.
I repeat question. Is there any scope of
Anyone can
gene
repeat the
editing?
question?
Satomi, Satomi, your voice is breaking. So can you please put your questions in the chat box if you cannot put in the Q&A box? Because for the panelists, I think we cannot put in the chat question
Okay,
box.
fine. Okay, fine.
Yeah. Chat box, can you please put in the chat box?
Okay.
Okay, in the
Or
meantime,
I mean, if yeah, of course, that's true.
okay, in the meantime, I have seen Sir Senthil among the panelists.
Yeah, yeah.
Sir, good afternoon.
Very good afternoon to all of you. And yeah, I was listening to Ravendran regarding his talk. I feel the high quality of research he is doing. It was really very nice. Ravendran, yes, in as you mentioned in your slides, the plant aspects and the microbial aspects you have, I mean, has a lot of potential. Can you, suggest a few things that could be done, for example, in medical aspects, like if we have to tackle a disease like cancer, what is the potential in UK? Any groups, research groups that work on it, and is there, a chance that we could discuss further on, in case if you are interested to venture into or put your knowledge into that field?
Particularly, I mean, initially, system was developed to use in the medical field, actually. And in Oxford and Cambridge, quite a lot of applications related to medical is going on, actually. I'm quite happy to, and also the plasmids, not only for microbes and not only for mitochondrial engineering. So it's a universal synthetic biology toolkit. So it's available in the Adgene. So you can purchase those plasmid and then you can use it.
I think now Dr. Tommy's question is there in the chat, box.
Yeah. So Tommy asked, is there any scope of gene editing in viral RNA like a COVID-19 CRISPR technique? Yes, actually, currently in US and UK, using a CRISPR Cas9. I think there are some groups who are using a CRISPR CPF1 technique to screen and knock out gene COVID-19. But we need to keep in mind the COVID-19 RNA rather than a DNA. So it can mutate quite rapidly. It's similar to AIDS. So still, researchers are finding of generating vaccines for things can apply to the COVID-19 also. The easily mutatedly. It's not stable. So it will take longer. Yeah, but there are techniques available using a Cas9 system to target the gene.
Okay. So no more questions from the panelists. Okay, I think they are clear enough with your
Is there any
presentation.
question?
I think no more questions. So we have come to the end of our rich yet fruitful 5-day webinar. Before we wind up the program, may I now call upon our Head of Department, Dr. Lehmang Iswali Ralte, Ms. Tete, to give a vote of thanks. Ms. Tete, please.
Yes, thank you. Good afternoon. As we have come to an end of this webinar, I, Dr. Lelma Izwali Ralte, on behalf of the organizing team of the 5-day international webinar on science and technology, mitigating current challenges, would like to convey a vote of thanks to all those who have contributed for the success of this webinar, directly and indirectly. I would also like to thank especially to our 5 eminent resource persons, Professor N. Senthil Kumar, Mr. C. Vanel Sanjima, Dr. Rita Zomanpui, Dr. C. Lalremrata, and Dr. Ravendran Vasudeva for gracing our event by enlightening us with their findings, knowledge, and opinions. We have gained so much from your presentation. It has been an informative and productive session on combating and mitigating the current challenges we face in our daily lives, especially in this time of pandemics. So a special thanks also to our principals and staffs for their enthusiastic support. I also thank the webinar organizing committee of our college and the Department of Botany for their support and coordinations. And also would like to thank Dr. Lalun Pluang Silo and Lerming Moy Goldi, who took their time amidst their busy schedule to be on the technical duty for the smooth functioning of this webinar. And also a heartfelt gratitude to our anchor, Dr. Lerem Siami Shaksir and Dr. K. Lerming Sangi for their continuous dedications. And I would also like to thank Mr. Liltin Puya, Department of Economics, for preparing the YE flyers and the YE certificates. And lastly, but not the least, I would like to thank all the participants for their active participations and interactions throughout this program and making this program a fruitful one. Thank you all.
Thank you, Ms. Tete. So with that, we have come to the end of our international webinar. So goodbye, everyone. Stay home, stay safe. Thank you. Have a good day.
Yeah, thank you. Thank you.
Thank you very much. Bye.